This video is part 1 of Chapter 7, Developmental Psychology for PSY 110S, the Psychological Sciences. In this chapter, we're going to cover development across the lifespan from prenatal development all the way to the point of death. Now, what you'll notice right off the bat is that we have a lot of territory to cover, and in most psychology programs that offer, a full major in psychology, there are going to be several courses in developmental. Some emphasizing early childhood, some emphasizing adolescence, some emphasizing adulthood and aging. In some cases, you'll have courses that focus specifically on types of development, like socio-emotional development, for example. So just understand that developmental psychology is a very broad field, and we're having to condense a lot of information into a smaller unit for an intro course. Developmental psychology, as defined by your authors, is the scientific study of biological, cognitive, social, and personality development across the lifespan. Developmental psychologists are primarily involved as academics in doing the basic research into these areas. Some developmental psychologists work in applied settings, but most are working in academia, conducting research. The chapter is going to focus on three major domains of development. First on the biological side, then on the cognitive side, and then on the social side. What you'll notice throughout the chapter is that, as with other areas that we've looked at in this class so far, nature and nurture have an interactive role in shaping development. While most psychologists today believe that nature and nurture can't really be disentangled from one another because they are mutually impactful. What you're born with in terms of your genetic endowment is going to interact with and be shaped by the environment you're in. And in fact, the environment that you're in can alter what nature has given you. So you'll notice as we move through the chapter, a lot of these theories, as well as a lot of the research that you're going to have described to you, involves this interaction between nature and nurture. We can divide the lifespan up, and I would argue somewhat arbitrarily, into stages. Understand that these are socially determined or culturally determined stages of life. There's nothing really essentially biological about these breakdowns, although in some cases you could say, yes, the prenatal stage has a clearly defined set of parameters. And adolescence tends to be tagged with the onset of puberty. But the rest are pretty much socially designed to describe a particular period of life. Not all cultures have the same breakdowns. The breakdown you see on your screen is very familiar to and most of the people in Western cultures, and particularly Western developed cultures that require a significant portion of childhood to involve schooling, this particular template tends to fit. Now, many cultures do not have that kind of structure built into early life. So there are cultures where schooling is not required, or it's not required of all people. And it's only a very privileged few who have access to such amenities. So in many cultures, and in our own culture in previous decades, and previous centuries, actually, we didn't quite have these sorts of breakdowns. Adolescence, for example, wasn't a defined period until well into the history of the United States. And just labeling that period from 12 to 18 or so really didn't evolve until compulsory education went beyond the point of the age of 12. So what we had was we needed some way of designing a label for these people who are not quite adults, but who aren't really children either. The young adulthood, middle adulthood locations in this model, in Western cultures, young adulthood is kind of bounded by getting your career kind of sorted and getting your family started. Then in middle adulthood, you're shipping your kids who are now grown off to make their own families, and you're getting ready for retirement. Again, these are very socially prescribed and agreed upon periods within the lifespan, but do understand that they are somewhat arbitrary. Now we're going to start our discussion with prenatal development, because there is a lot that happens before people are even born that can have an impact on their development later, after birth. So we'll start with just a little journey into prenatal development. So what's happening in this animation is that you get sperm entering in through the vaginal canal, making their way through the female reproductive tract to eventually meet up with an ovum, an egg cell that is ready for fertilization. Obviously, a lot more sperm are being delivered than are actually necessary, but that is a way of kind of hopefully ensuring fertilization of that mature egg. Now here you're looking at a mature ovary, and an egg cell is going to be delivered into the fallopian tubes. And then once that occurs, it will migrate somewhat slowly down the fallopian tubes, where eventually it will encounter some sperm. These little cilia along the fallopian tubes help to keep the egg moving on its little journey down the fallopian tubes. They have this waving, undulating kind of motion. The sperm are going to swim up and encounter the egg, and eventually a lucky sperm is going to be able to fertilize that egg. Most of them will not. So after fertilization, then the duplication and expansion and growth of that fertilized egg begins. And at the point when this fertilized egg, which is already starting to do dividing and growing, is still migrating to the uterus. Once it arrives at the uterus, in the female reproductive tract, that uterus at that stage in the menstrual cycle will be all prepared with a very nutrient-rich lining, which, if pregnancy doesn't occur, if that dividing brand new fertilized egg does not implant into the lining of the uterus, then that lining will be shed during menstruation. Now in this case, you have implantation, where that rapidly growing zygote is implanted into the uterine wall. Then growth and development continues at a fairly rapid pace, although for most previously pregnant people, not fast enough, but there you have it. So, we all start out not looking very human, but over the course of time, over development, we start to develop all the structures that will eventually turn into our skeleton and our organs, and all of the basic pieces, parts that make us what we are. So conception is defined as the moment, the time when a sperm penetrates the outer membrane of the ovum, carrying the genetic complement from the male of the species. When the egg and the sperm combine, there's a complete set of genetic instructions. So you have half of the instructions from the father, half from the mother, and that's what produces a zygote. Every cell for humans under normal circumstances is going to have 23 pairs of chromosomes, including one of each pair coming from the male and one from the female. A zygote, that term refers to a fertilized egg that comes from sperm and egg uniting through human reproduction. All the other cells in the body are going to develop from that single zygote. So during that process, you're going to have lots of differentiation and separation. The genetic information coming from the male and the female are going to dictate the nature of that process. Now, sex determination, on the one hand, seems very, very straightforward and simple. On the other hand, it's not as simple as you think it is. In general, so most of the time, when you have on the 23rd pair of chromosomes, it's going to determine the person's biological sex. Female humans typically have 2 X-shaped chromosomes. And why am I saying typically? Well, you can have a person who appears externally in terms of their external sex characteristics to present female or to present feminine. They may or may not have 2 X-shaped chromosomes. The same, I'm going to say the same thing about males being XY. Not all people who appear biologically male externally will necessarily have an X and a Y. There's a fair amount of variation there. But generally speaking, most commonly, females will have 2 X chromosomes. Males will have 1 X-shaped chromosome and 1 smaller Y-shaped chromosome. The Y chromosome is what triggers the development of male sexual characteristics, external characteristics, internal characteristics that are consistent with being male. So having testes as opposed to ovaries, for example. There are variations, as I mentioned before, and these lead to what are referred to as intersex designations. So not all people have an X, X, or XY. You can have multiples, so there can be triple X. You can have singles. You can have a single X. You can have XXY. You can have XYY, and so on. Now, typically, these variations come with some consequences in terms of specific changes to cognition, to motor skills, to physical appearance, and so on. Some are not obvious in terms of what's going on chromosomally until puberty, where the changes hormonally that are expected either don't occur or changes that were not expected do occur. Now, you would have to take, further courses exploring some of this to learn more about those various intersex conditions. Twinning is always possible, and multiple births are always possible. Identical or monozygotic twins, that occurs when you have your initial zygote basically splits early in development, and you have two clusters of identical gene sets. These clusters become identical twins. They're from the same zygote. They're just, splitting in two. So you have two individuals with the same genetic complements. Now, does that mean that they are going to be 100% identical after the course of pregnancy and even after birth? No, not necessarily. In fact, when you look at identical twins, you can see numerous, sometimes subtle, sometimes not so subtle differences between the twins. And what we refer to some of these differences as is epigenetic changes. So twins, whether they're identical or fraternal, so-called dizygotic twins, they do not always experience the uterine environment in exactly the same way. It's not uncommon, for example, for there to be one twin that's slightly physically larger, and sometimes described as a more dominant twin biologically, and the other twin being somewhat smaller. There are even arguments that that larger twin has some more protections against sort of environmental insults, where the smaller twin may be more vulnerable to some of those impacts. Fraternal twins or dizygotic twins are, that happens when you have two eggs released in the same cycle and both are fertilized. So you have somewhat different genetic complement, roughly equivalent to what you would have with siblings. It's just that they're carried in the same pregnancy. So in dizygotic twinning, you have two separate eggs, two separate sperm, and the creation of two separate individuals. In monozygotic twins, you have one zygote coming from the same fertilized egg, and it splits in two, creating two individuals with the same genetic complement. In prenatal development, it's typically been broken down into three stages. You have germinal, embryonic, and fetal stages. The germinal stage starts with the formation of the zygote and ends at about 2 weeks, when implantation into the uterus takes place. The embryonic stage runs from 2 weeks to 2 months, and during this time period, most of the major structures of the body have gotten their start and can be visualized under strong magnification. In the fetal stage, 2 months to birth, you have further development of all of the structures and organs. We make a shift from talking about an embryo to talking about a fetus. So it goes from zygote to embryo to fetus. And during that time period, you have very, very rapid growth. All the body structures and organs complete their development in a pretty predictable order. During that pregnancy, you have the development of the entire skeletal structure, the development of eyes and other sensory systems, the development of all of the organ systems, and the brain. There are lots of things that can impact prenatal development, and it includes, complex interaction between genetic and environmental factors. Development is influenced by, first, the genetic code, but it's also impacted by the environment, and the most direct environment is the mother that's carrying that zygote, and then later embryo and fetus. So anything that the mother consumes, anything the mother is exposed to, including stressors, can, at least theoretically, have an impact on the growing and developing zygote, embryo, fetus, baby, whichever term you prefer. Also, things outside of mom's body, including the physical environment that surrounds that person, whether there are toxins in the environment, stressors in the environment, all of those things can have an impact on that growing, developing baby. Things that can be dangerous to or can cause negative impacts from the environment are referred to as teratogens. These can be agents like drugs, viruses. It can be diseases like German measles. It can be physical situations like not having enough nutrition. And, I would add there all kinds of kind of cultural situations, like, are you in warfare? Are you in living in a space that is very filled with strife and insecurity? Are you unhoused? Those sorts of things. All of those can be teratogenic agents, meaning that they can have a negative effect on the growth and development of that being prenatally. Now, you're probably well aware of the warnings out there against doing such things as drinking alcohol, smoking tobacco, taking illicit drugs, and even some prescribed and over-the-counter drugs during pregnancy because of the risks to that developing child. One example, if too much alcohol is being consumed on a regular basis during pregnancy, one outcome that can happen is something called fetal alcohol syndrome, which can have some pretty wide-ranging impacts cognitively, socio-emotionally, behaviorally, and in terms of motor development. So, again, you're probably well aware of many of these arguments because we have had a lot of public messaging about the consumption of alcohol, tobacco, and other kinds of drugs and toxins for many, many decades now in U.S. culture. Now, the likelihood that there will be health risks to a growing, developing fetus also can be impacted by the age of mothers, and we're starting to find that it can also, the age of fathers can also matter as well, although that's been studied somewhat less. We do know that mothers who are very young, who are younger than 15, that tends to be associated with health risks to the fetus, and women who are over 35, there appear to be health risks as well. Now, as I said, we're starting to have research findings that suggest that there are also issues that are associated with advanced paternal age, so the age of the father, that can also impact health risks for a growing, developing fetus. Other kinds of risks are anything that contributes to prematurity, so being born before being full term, and anything that can impact the birth weight of the baby. And that's lots and lots of factors. In terms of motor development, one of the first things that parents and also medical professionals are going to be looking for is to see that babies come into the world with an intact set of reflexes. So, right away when babies are born, the attending physicians are going to check for very specific forms of reflexes. Most of these reflexes are related to survival, so things like sucking and rooting are designed to help with feeding, so that baby is prepared to either nurse from the breast or to suck on a bottle. Some remain after birth, like as soon as that baby is born, the breathing reflex should be triggered so that the baby can breathe on its own. So that's going to remain. Others are going to disappear. The Babinski reflex, which is when infants will fan their toes upward when you touch their feet. That's like a leftover reflex from sort of our evolutionary history of needing to use our feet to grab onto our fast-moving moms. So, if you see chimpanzees and great apes in the wild, their little babies are often, mom is on the move, and they are hanging on to her for dear life, and they're using hands and feet to do that. That reflex, opening up those toes when they touch something, is associated in some ways with that old need to use your toes that way. That reflex disappears over the course of early development. The grasping reflex, same idea. It's often startling to people just how strong the grip of a little tiny newborn can be. We often think it's super cute when they grab a hold of our fingers and think, "Aw, they love us, and that's why they're grabbing our fingers." Well, not really. They would grab anything, primarily because, again, in our evolutionary history, it was best if you just grab on and don't let go because your mom's on the move, and that reflex will go away over time. Now, developmentally, and this is one of thousands of images like this that I found online, just to give you a visual of the milestones that parents and providers are going to be looking for as kids move through the first 16 months or so of life. From 0 to 1, you want to start looking for that baby to be able to lift its head. When babies are first born, they've got pretty much no control over their head. Over the course of the first couple of months, they start to develop more control over their neck muscles and more strength in those neck muscles so that they can lift their own head and hold their head still while they're sitting up, and even when they're being held. From 2 to 4 months, you have babies being able to push their chest up and use their arm for support. By the time they hit 4.5, 5 months, they should show signs of being able to roll over. Some babies do that earlier than others. Between about 3 and 6.5 months, you should be able to see the baby being able to support weight on its legs. From about 4.5 to 8 months, babies should be able to sit without support. By 10 months, they should be able to stand without support, or with support rather. During that same period of time, by 10 months, they should be able to pull themselves up to stand. You just have a broader bar here, anywhere from around 7 months to 12.5 months. You should be seeing babies, toddlers at this point, walking using furniture or people for support. Between 10 and 14 months, being able to stand alone, and between about 11 and 14 months, being able to walk alone easily. Now, the size of the bars here on this graphic show that there are individual differences. Some kids hit their milestones a little later, some kids a little earlier, and depending on whether they hit them early or late depends on a whole bunch of factors. Some are genetic, some are environmental, most are an interaction between the two. Visual development is something you can track across early post-birth development. At birth, vision is our least kind of finished sensory system. Our babies, when they're born, just really can't see all that well. Their visual acuity is pretty crappy, 2400 to 2800. I don't know if that's officially considered legally blind, but it's pretty darn close. At about 2 or 3 months, kids start to be able to see color, although not the full range, but pretty soon that starts to get clearer. Within the first year of life, so by the time you end that that 12-month period, visual acuity gets far, far better. Now, when you think about it, and this is a graphic that I found that shows, as newborns, you're not seeing any color. By 4 weeks, you start to get some gradations of color added, but they're still kind of grayed out. By 8 weeks, a little more. 3 months, a little more. 6 months, a lot of color vision. You still got some blurriness on the edges. By the time you're in adulthood, tons of acuity plus color. So you move from very, very blurry to very, very clear in a fairly short period of time. Infants, brand new newborns, they're designed really, since they're not going to be moving around their environment, having a fully formed visual system isn't all that important. So in comparison to, say, a newborn baby horse, they have to hit the ground when they're born, stand up, nurse a little, and be ready to run after their mom, because they're a herding animal. Vision-wise, their eyes are pretty well developed, and they need to be able to see things that are moving in their environment. Human infants, not so much. We can't run away from anything. We can just kind of lay there. We have to be picked up. We have to be taken care of. So infants, pretty much their best vision is about as far away as an adult would hold that baby in their arms. So, about a foot away. And they're most attuned to picking up facial features in the broadest sense. So if their eyes are really, really blurry, within that first, you get to 4 to 8 weeks or so, there's enough acuity there that you can make out the rough features of the face. And that's really the critical time during which that sort of attachment starts to build. Now, one of the ways in which developmental psychologists study sensory development in infants, because you have to be pretty crafty. If you're a developmental psychologist, you have to be pretty crafty because infants aren't talking yet. And even if they were like 2, 3, 4 years old, what they're going to say to you may not have the language, the sophistication to give you the answers you want. So as a developmentalist, you're going to have to use some pretty creative techniques to study the development of sensory and perceptual abilities. One of the tasks that's been used is something called a preferential looking technique, which involves studying visions in infants where you're presenting visual stimuli. Usually it's 2 visual stimuli displayed side by side, and the researcher is going to record how much time the infants spend looking at something that's presented in their visual field. So you present something. So in this graphic that I've shown you here, you present them something. If it's a new stimulus, babies will look at it longer. If it's an older stimulus, they'll look at it for less time. The assumption is that if infants look at one side of a 2-image display for longer, it's assumed that there's a preference. Now, on the one hand, if you have a novel image, there's a tendency for infants to look at those new things longer than things that are very familiar. They will eventually converge because that novel display is going to eventually no longer be novel. But what you're looking for is to see whether or not there's this preference for the new thing, the new thing that is now interesting in terms of what's being looked at. Now, habituation happens when the infant doesn't really respond with much interest or preference to a stimulus once it becomes familiar. So on the left-hand side of this graphic, an infant in the first 2 2 presentations, very interested, looks at it for a long time, but then over the course of the 3rd, 4th, 5th, and then down into 6, 7, and 8 presentations, not so interesting anymore, actually pretty boring. When you add new elements, so you have the bottom line where it says familiar display, you show that image, they're going to continue to, they'll acknowledge it but ignore it. You give them another new thing to look at, they're going to pop up, look at it for a while, and then habituate to that as well, going down to baseline. Infants do tend to look longer at new things that they haven't experienced before, and what we're doing when we present this kind of task is inferring that they have to be able to tell the difference between new and not new. And that's, you know, for little infants, that's a pretty big feat. Now, it has been suggested that even in utero, so prenatally, if you're not talking about vision, but instead talking about hearing, since infants can hear, I mean, it's by far muffled, but infants do show preferences for voices of their caregivers, particularly their mother. They show familiarity with those voices and preference for those voices in terms of their activity level in utero. Also, another kind of measure that can be used is, in addition to how long they look at an object, they can also measure using a specially defined pacifier, which records how intensely the baby sucks on it. They tend to intensify their sucking when they are shown a new stimulus. When I've seen this on video, what can happen is the new stimulus is presented, there'll be this one moment of pause on the sucking and then really fast sucking. So it's in some ways excitement, interest being shown in that way. So again, we have to be pretty crafty by either documenting how long infants gaze at an object or how intensely they suck on an object. Since they're pre-verbal, they're not talking yet, and they can't move around their environment as little infants either. Another element of visual perceptual development is the development of depth perception. And really, this isn't necessary. It's not necessary for you to be able to understand depth visually until you're going to be moving around your environment. So what some researchers, Gibson and Walk, designed was a task that you can see in this picture called the visual cliff. And essentially what the apparatus is, it creates the visuals, so the red and white checkered flooring separated by a white line and then plexiglass that covers that same red and white checked pattern, but the red and white checked pattern that's hidden underneath the plexiglass is about 3 feet down or 2 feet down. Nearly all infants, by the time they get from 6 to 14 months, so they're crawlers. They're crawling, they're starting to walk, they're moving around their environment on their own. As a result, it'd be really nice from an evolutionary standpoint if they would be afraid of walking off a cliff. That would be a good thing. So in terms of development, without a lot of help from grown-ups, it's almost like a switch gets flipped where this now toddling, crawling baby needs to respond to the environment to protect itself from falling. So what physicians are going to be looking for in terms of testing, or developmental psychologists are going to be looking for in terms of testing, is during that critical period, are babies showing a refusal to walk across the plexiglass toward a caregiver? So as you can see in the image, there's that open hand. what they do is position the toddler at one end of the visual cliff, and then the parent will very excitedly call the toddler and encourage them to come to them. If they stop on the white lines and show distress and unwillingness to go across the plexiglass, what that means is that they perceive depth. In further research, Adolf and colleagues found that there's an interaction between that sort of evolutionarily developed need to perceive depth, but it also relates to learning experiences. The more mobile kids are, the more time they're given to explore their environment, the more refined their depth perception becomes. So as they get more experience with the physical world, moving through that world, it can influence the expression of a fear of heights. Now, there are tremendous individual differences. Some babies and toddlers will develop a healthy fear of heights, an appropriate level of fear of heights that starts to fade a little bit as those children get more sophisticated in their locomotor skills and their experience with the world. Some babies and children don't develop solid depth perception and therefore don't develop solid fear of heights. So there are individual differences there. Now I have a love-hate relationship with this graphic, this illusion. Infants, from very early on, as soon as their vision starts to be just a tiny bit less blurry, they tend to show preferences for faces. And even just organizations of shapes that have a face-like quality to them are preferred. And very, very small infants, very young infants, can tell and show distress when those basic features are messed up. So in the image that you see here, it's called a Thatcherized face. So the images of Margaret Thatcher, the late Margaret Thatcher, who was the Prime Minister of England. And what you do in a Thatcherized face is when you take parts of the face and flip them. So adults readily detect these changes in face patterns when the faces are viewed upright. So if you look at the bottom two images, the one on the left is the natural face of Margaret Thatcher. The one on the right, her eyes have been flipped upside down and her mouth has been flipped upside down, and she looks pretty darned horrific, right? Really pretty terrifying. So adults pretty readily recognize that. Infants, not so much. The faceness organization is still present, but the fine-grained parts of, like the upside downness of the eyes, the upside downness of the mouth, is not something that infants are going to be able to determine. The ability to make sense of the configuration of upright faces, so, the relationship between the parts of the features. So two eyes spaced relatively close together, separated by a nose, with a mouth underneath it. That configuration is likely inborn because we can show, researchers can show that, as soon as infants can make any kind of image, especially close up, they can determine, when they're in the right positions, when they're in the right order. And they get more sophisticated with that over time. Leo and Simon created the Thatcherization effect that's created by rotating the eyes and the mouth 180 degrees. In the bottom part of that image, the bottom right image, pretty awful looking. What they found was that newborns, they understand that things should be in a particular kind of structure. Over time, they will start to develop the kind of horror effect of that bottom right image. If you flip the two images upside down, you don't get the same negative impact for adults. So in the top row, the right-hand top right image, not so horrifying. Part of that is because our brains are processing eyes that are in the appropriate orientation for our own perception of them, because we're really looking for information from those eyes. Infants aren't quite ready yet to make inferences about mood, for example, or intent, or even emotion coming from the face. So we're less horrified by the upside down Thatcherized face than we are by the right side up Thatcherized face, because the right side up one, those eyes look really demented, and that mouth is very frowny and is showing teeth. So what we're getting is a bunch of confusing messages about what this face conveys. Understanding emotion and intention from the face is fairly sophisticated. So while infants know where the eyes, nose, and mouth should be pretty much from the start, the nuances are not quite present yet. Hearing, it develops pretty strongly while we're still in utero. So before birth, babies can hear. When they're exposed to auditory stimuli from the outside world, they respond to it. Newborn hearing is much more fully developed than vision. In fact, babies are pretty sensitive to sound, as anyone who has accidentally woken a baby up can attest to. They can, under testing, tell the difference between their mother's voice and the voices of others. They show a preference for mom's voice, and they show preferences for the voices of others who spend time around mom. So they can discriminate between mom's voice, other caregivers' voices, siblings' voices, and those of novel strangers. By about 6 months, infant hearing is about the same as adult hearing in terms of precision and range. In childhood and adolescence, you get continued improvements in perception, continued improvements, especially in the domains that are connected to language. So over the course of childhood, from birth, babies are pretty much attuned to the ranges of sound that are associated with speech, and they are prepared to pull in as much information as possible that's going to be related to their development of language. And that concludes part 1 of Chapter 7.